Recording. So I wanted to thank everyone so much for joining us at our quarterly PMedic seminar. It's my pleasure to introduce our OHSU speaker, Dr. Christina Tanyong, and Bobby Jo Webb-Robertson will introduce the PNNL speaker, Dr. Sarah Gosselin. So before coming to OHSU, Dr. Tanyong received her bachelor's degree in genetics from the University of Western Ontario in Canada and her PhD in medical genetics from the University of British Columbia, where she worked for multiple years as a cancer researcher. Christina Tanyong has now been at OHSU for 10 years as the scientific director of Brian Drucker's lab. She's worked on numerous large collaborations spanning academic, government, and industry partners, and also works as the OHSU Heme Scientific Liaison with our AstraZeneca Partner of Choice program. She is currently engaged in research studying late and early resistant mechanisms of resistance in acute myeloid leukemia and is part of OHSU Knight Cancer Institute's Beat AML project, a comprehensive platform for analyzing leukemia cells, their sensitivity or resistance to drugs, and the medical outcomes of more than 1,000 people with AML from across the country, which led to the data that will be presented here today. I'll hand it over to Bobby Jo.
Actually, I believe Jason just joined us. Jason was going to introduce Sarah
Oh,
today.
right. Sorry. Thanks.
Yeah, sorry.
I don't have as many titles as Christina. I don't know. It's a tough act to follow.
So I'd like to welcome Sarah Gosselin. Sarah has been at PNNL for 3 years now, almost. Sarah is currently a team lead of software, and their name just changed. The team that she leads just changed. So it's software and analytics. No.
Software and algorithms.
Oh, so close.
Analytics is too big.
I would do yeah. So she has worked in network biology. She has a computer science background, worked in network biology, worked in cancer and rare diseases, where she is working now. And she has been working on the PTRC since she got here, and has really hit the ground running when she got here, and has ably taken over the bioinformatics portion from the PNNL side of the AML project.
Awesome. Thank you, Jason.
And I didn't give any of your pedigree because I actually don't have it in front of me, so I don't want to like mess it up. But you can certainly mention that if you want.
I can just say that I also did my PhD in Canada, so we have a strong northern contingent here, even if I don't have quite the accent.
Where did you do it, Sarah?
McGill.
Oh, okay.
My master's and my PhD, yeah.
We'll have to talk about that later.
Yes, yes. Yeah, so I have lots to say about Canada, and especially if you talk to me about snow. Okay, cool. I think I'm going to go ahead and get started then, if everyone's okay. Awesome. Yeah, so I'm super excited to present this work with Christina because we have been working really side by side in the trenches for the past 3 years on this project, on these projects. And Christina's obviously been working on AML for a lot longer, and she'll describe that work. But it's really exciting work. And the title is Mapping the Proteogenomic Landscape of Acute Myeloid Leukemia to Enable the Prediction of Drug Response. And I think it really highlights the proteomics work that PNNL knows and loves, and which has sort of become our bread and butter, as well as the computational stuff, which is quickly becoming our bread and butter. I'm going to go ahead and say that. And also the really deep bench that OHSU brings to the table as far as acute myeloid leukemia. Again, we have lots of projects ongoing that's been really exciting. There we go. So I'm going to talk about this word landscape a lot. And so I like to give this slide. A lot of us have seen this before, but it really kind of harkens back to Waddington's landscape, where Conrad Waddington liked to describe a cell as a very primitive state where it could become absolutely everything it wanted to be. And through a series of epigenetic changes, you will then have the cell just differentiate itself to become a skin cell versus a bone cell versus a, I don't know, eyeball cell. Okay, I know there's no such thing as an eyeball cell. And so we kind of take that same metaphor when we talk about cancer in this case. And here we're not talking about a cancer cell, but actually a tumor. So here we have a tumor which starts as a skin cell, right? And then actually goes through a series of changes as it becomes cancer. But you know, while many of our skin cells look the same, our skin cancers are actually vastly different. So as my, again, skin cell, I'm going to keep going with that metaphor, I guess, evolves and becomes cancer, it looks different than another patient's cancer cell. And this is manifested through various genetic mutations that are distinct from other people's tumors, as well as transcriptional changes and protein signaling changes. And so when you're treating skin cancer, you're actually treating a suite of a whole collection of diseases that all sort of fall in different places on this landscape. And as a result, treating the patient's disease is actually like treating thousands upon thousands of diseases across this landscape. And so you have on the right here, I'm going to, this tumor that is quite resistant to a drug response, as shown by a dose response curve that kind of is shifted to the right with a high area under the curve. And then you have this tumor on the left, which is quite sensitive to the drug. And so, one of our dreams in cancer biology is to try to identify not just, maybe not the exact drug for each tumor, but also the drug that can actually shift a patient's tumor from that resistant phenotype back up and around more to this green guy here that is sensitive to the drug. And so the way that we do the mapping, to this landscape is through omics data collection. We do genetic mutations, transcriptomics data, proteomics data, and phosphoproteomics. And we do this with hundreds of patients with this idea that we can then map the patients to this landscape, collect the drug profiling, and then do the wizardry that will allow us to really enable the drug prediction response, which is basically all I say in this slide. We find a good system, a way to evaluate the disease, both with omics data and a way to test it via drug response, which is what I'm calling the model system here. And then we really scale it to sample all parts of this landscape. And then we try to model the approach of how different points in the landscape give rise to different drug response, which is essentially sort of the outline of this talk. I'm not going to talk about the model system development as much, but really the mapping of the landscape, which Christina is going to talk about with her Beat AML work, and the work that she's done with many collaborators at OHSU. And then the prediction from the proteogenomic landscape, which I will talk about in detail after Christina's done. So with that, I'm going to pass it over to Christina, and just let me know when to advance the slides.
Yeah, sure. Thank you, Sarah. So I guess I'm tasked with giving a broad overview of acute myeloid leukemia, which is the model system that we're looking at, and then going into the details about the large cohort that we developed here at OHSU, along with collaborators across the country. And I do want to make the caveat that I'm speaking on behalf of a very large group of people here at OHSU that pulled together to make the Beat AML cohort happen. It was definitely not my, it was led by Jeff Tyner and Shannon McWeeny, and then all of us, the sporting crew, for their vision of getting this done. It was a monumental task. So, it really focused on acute myeloid leukemia, and we chose this as one of the main focus of our project because of the poor outcomes that we observe in patients with AML. It's a 25% 5-year survival rate currently, and that hasn't changed very much over the last few years, even though we've identified a number of new drugs that are available for these patients. 21,000 diagnoses a year in the US and 10,000 deaths. And over on the left here in the pie chart, you can see this genetic diversity, the mutational diversity that is observed in patients with AML. which makes it a very heterogeneous disease and a very difficult disease to treat with one hammer, so to say. Really, we need to understand the landscape of AML and understand how each individual patient is different and how they respond to various drugs. And that was really the driving goal for the Beat AML program, which we developed going back to 2012, really. Next slide, Sarah. So in 2012, we developed a program called Beat AML with the help of the Leukemia and Lymphoma Society and support of the Knight Cancer Institute. And we call this Beat AML 1.0 because of there are a couple, and you'll see in the talk, there's a couple different waves, but there's also currently a clinical trial called Beat AML. It's a Beat AML Master Clinical Protocol clinical trial, and that is a little bit confusing to individuals. So this is the research program that I'm going to be talking about today. The overall goal was to collect over 900 primary AML patient samples. These are peripheral blood or bone marrow from patients. And in order to do that, we created a consortium of over 11 academic medical centers from across the country. And they would send samples to OHSU as the coordinating center, where we would process the sample for derivatives that we could use for genomic analysis. We would plate the sample on drug plates to determine functional response. And we had a large number of samples that were profiled for the immune microenvironment. And this was led by Dr. Evan Lind. All of these data kind of came together and were analyzed by different computational groups, with Shan McGuiney and Dan Bottomley being the major drivers here at OHSU that led that effort. And this information eventually gets back to the lab, where we do additional wet lab validation to verify targets that are identified, or do really expand out the initial findings from this data. And our overall goal is to really have some information that we can go into clinical trial development with, either through an investigator-initiated trial or through approaching different pharmaceutical collaborators to put clinical grade drugs on our drug screens, and then moving that into clinical trials with them in industry-sponsored trials. So our first paper came out in 2018 in Nature, and it really looked at approximately 600 patient samples at that time. So next slide, Sarah. Just to give you a really quick overview of this functional drug testing that we performed, and this was really the unique part of the Beat AML program. We isolated leukemia cells from bone marrow or peripheral blood from AML patients using a Ficol gradient, and then arrayed the cells into 384-well plates that were had different dose gradients of drugs, and from a wide variety of drugs, kinase inhibitors, different types of drugs with different mechanisms of action. The cells were then cultured for 3 days, and then cell viability was assessed using a colorimetric assay, like a MTT assay. And then we could determine the IC50 or the AUC of the drug response in that patient sample for a number of different drugs. So this is when we talk about the ex vivo testing or the functional drug testing, this is really what we're talking about. That is really unique for our samples. Next slide. So this initial cohort, we really had a lot of different data, exome sequencing, RNA-seq, drug response data, and a very deep amount of clinical annotations for these samples, which in the end became a really valuable data set, which was led by our senior data manager, Nicola Long. And we were able to use all this data in various ways, either integrating the drug response with mutational analysis, the drug response with expression to find gene signatures that associated with drug response, more complex analyses where we could actually integrate the different data types, including expression, mutation, and a drug response, and identifying different modules that would be actually associate with different features. And then we could also use the clinical data and see if there were certain clinical features that would associate with drug response. So these are the types of analyses we're able to do with this. Next slide, please. And a lot of this data was made, all of this data was made freely available in a web-based browser that Shannon and Dan Bottomley, Beth Wilmot, Eric Segerdal, and Libby White put together. It's called Visome, and really was able to, the goal was really to democratize the data. So you can go into Visome. There are a number of data sets here that you can go in and start to play with visually in using these different modules. And I encourage you to go take a look at this because it really is quite interactive tool that you can kind of look to explore the data. And our group uses it all the time to just identify new features. You can also download figures, and you can also download the data set from here as well. So that's one of the unique features of our data set. Next slide. So from this work, we've really been able to have broad impact over and above what we published in the Nature paper. So it continues to bear fruit, really markers of mechanisms of drug response, multiple papers. We've identified novel recurrent oncogenes that were not described before. One of the features of the data set is that we collected skin biopsy controls from the patients, and we're able to isolate germline DNA. And this was mined by Anupriya Agarwal and Fan Yang, and Nicola Long did a lot of work for this, really looking at the germline predisposition for AML, which is really interesting. Evan Lynn's lab with Adam Lamble did a really great paper on T cell suppression, using the samples and analyzing by CyTOF and other ex vivo assays. We had clinical outcome determinants that were analyzed. We sent samples, DNA samples for methylation array sequencing, to be able to look at the epigenetic landscape of the samples as well. So smaller cohorts of this larger cohort. So really the broad impacts of this data are ongoing, and we continue to mine and use this data, as do all researchers across the world. Next slide, please. And this is the one you can click through a number of times for me, Sarah. So that was that's great. The first paper that came out. We have since this year published a larger paper that encompasses not just waves 1 and 2, which was that first cohort of 600 patients. We added on waves 3 and 4, another 300 patients, and then harmonized the data across waves 1 to 4 to and summarize them in the cancer cell paper that came out this year. And of course, we had the very similar kinds of data types that we were using for this, but just a larger data set. Really interesting integration of the data in this particular paper. And I'm going to show some vignettes, but over to the right just shows you some of the approaches that were used in this paper in a graphical format, really. And I'll go through those one at a time very quickly here. So next slide, please. So there was really the question of, well, how concordant is the data? High concordance of both the genetic and drug response and expression profiles. And I'll show a couple of examples of that next. I think it's the next slide. So on the left, you'll see waves 1 and 2, and on the right, you see the waves 3 and 4, and very similar mutational frequencies observed, clinical outcomes, and drug sensitivity. Next, click one more, Sarah. And very high concordance when we do some analysis looking at drug response versus mutation or drug response versus expression analysis. So going forward, using the entire data set, we have a very concordant data set that we are able to use for very robust analyses. Next slide. I'm not going to go into this too much, but suffice it to say that we also had a number of new mutations associated with drug response and outcomes. And this is really because we increased our N in this very large data set. So we were able to really have some more robust statistical analysis of these associations. Next slide. I'm going to go into this a little bit, but Shannon's group did a lot of really interesting analysis looking at cell type maturation state. And I will go and explain that a little bit more because it is integrated with what Sarah is going to talk about. So next slide. For this cell state, it was really taken from a paper by Van Galen et al. In 2019, where they really looked at single cell RNA-seq data on AML patient samples and broke up the identified 6 different signatures for different cell states. And then what Shannon's group did was to take those signatures and then map it back onto the bulk RNA-seq data and then classify each of the patient samples falling into one of these cell maturation state signatures or within our Beat AML cohort. And that allowed us to really look at really interesting correlations with cell state. Next slide. We are also able to look at the correlation of cell state with drug sensitivity. And next one. And this is one of the vignettes out of that Cancer Cell paper, which looks at really the correlation of drug sensitivity shown in red or resistance shown in blue for different drugs across the different cell states. And there are a couple drugs that really pop out here, really panobinostat and venetoclax. So panobinostat is over to the left, and you can see that it has is very sensitive on the more mature, more differentiated cells, whereas venetoclax is more sensitive on the more immature or progenitor-like cells. And then, other drugs fall into these, similar to those 2 types of drugs can fall across that spectrum. So we talk about this in the work that Sarah's going to propose, and we look at a couple of these drugs as well. So I just want to kind of point that out. Really, a lot of the drugs show some kind of segregation of response based on cell maturation state, and that's a big theme in the AML world right now. Next slide. This cell state association with drug response has been corroborated independently by other groups such as John Dick's lab at Princess Margaret Hospital in Toronto, really showing a very similar thing that different cell states associate with the drug response. So a very common thing that's happening right now. Next slide. And lastly, all of these data can be integrated together, not just 1 or 2 data types, but really using a modeling approach and the outcomes data to really extract novel features out of the data. If you could go to the next slide. I'm just going to show one example in the next 2 slides, and I'll be completed my section, but this was a really interesting part of the data analysis that uncovered novel gene expression features that correlated with overall survival in our data set. So on the left, you see a heat map that really looks at these sort of different modules or even cell states that associate with, I have them boxed up in the corner here, the overall survival. So module 9 and module 3, which are a combination of different data types together, driving the association in a module, is shown to be associated with overall outcome. And then when you drill down into it, module 3 is highly correlated, has one of the strongest predictors for overall outcome. And if you go to the next slide. And when you look at what is in module 3 that could be driving this strong association with outcome, it became very apparent that a single gene called PAIR1, which is a platelet aggregation receptor, really drives the effect of PAIR1. I mean the module 3 prediction. And when you compare pair 1 alone, just as it's by itself, and take a look at its ability to predict survival probability, you see that it is very significant. And it's very similar to one of the better known gene stemness scores called the LSC17 score, which is associated, which has a high predictive ability to determine risk determination for leukemia or AML patients. So the single gene is very similar to this 17 gene score. So really interesting. We're uncovering a lot of information in biology about pair 1 right now as we're fascinated. And I think that's something that we want to look into with the proteomics as well. So I think that's my last slide, Sarah. You can take it away from here.
Awesome. Yeah. So I think, to sort of wrap up what Christina has showed, I think, what she showed is that, through this very careful collection of data, analysis, and harmonization, we've really been able to have this very rich data set that has, done a great job of surveying the AML landscape, both with estimating the patient cell population and with biomarker prediction and hopefully new treatments. Oops, that was not supposed to be animated. So now I'm going to move on to what we've done with the protein data, which is, sort of what we do. And I think this is a subset of what Christina showed earlier of the waves 1 through 4 data that's been harmonized. And from these patients, as part of the Clinical Proteomic Tumor Analysis Consortium, Karen Rodlin and Jeff Tyner collaborated and wrote a grant to collect 210 patients from these samples and analyze them via proteomics and phosphoproteomics. Ignore the 38 patients for now. To really, and so that with this idea that if we combine them all, we'll actually get a data set with proteomics, phosphoproteomics, transcriptomics, and genetics. And so, and functional screening. So this is essentially what we have. We have 210 patients with the proteomics data due to sort of, and they all have some level of ex vivo drug screens, the plates that Christina showed here, with the bar plots indicating, sort of the longer the bar plots, the more drugs that we have for that patient sample. We have 159 patient samples with RNA-seq and 177 with WES, with 76 of those patients having a FLT3 ITD variation, which was somewhat selected for, which is why the numbers are a little off, so that we could actually really capture that mutation with more statistical power than what would sort of naturally be occurring within a 210 patient population. So the first thing we did with this data is try to cluster all the omics data to identify what kind of subtypes were existing. So this is, again, instead of doing that supervised approach with the single cell analysis, we decided to do an unbiased approach using clustering. And so for this, we combined the transcriptomics data, the proteomics data, and the phosphoproteomics data using non-negative matrix factorization to identify 4 clusters. And this is a pairwise similarity between all the patients. It's 159 that have all the data. And so what this shows here is that we can actually choose any number of clusters with this non-negative matrix factorization, but we wanted to choose the ones that were most robust. So we took these, we redid our analysis using some randomization and different starts to see how frequently these clusters were put in this, these patients were put in the same cluster. And so if you see all red, that means they were put in the same cluster 100% of the time. If you see lighter colors, like the orange or the blue, it means that sometimes the algorithm assigned these patients to different clusters. So we wanted to have the picture, we have a lot of these heat maps, we wanted the one that showed the most red and the least amount of orange and light blue. And so the colors on the left indicate what subtype the patients were assigned. And the colors are here, and I'm going to talk about these subtypes a lot. So orange is 1, purple is 2, green is 3, and yellow is 4. We mapped some clinical variables along the top and did some enrichment statistics, but it wasn't super striking, statistically speaking. We had a slight enrichment of FLT3 ITD and subtype 2, but as you can see, it's still kind of scattered throughout, and I'll get to this later. We also have some higher FAB scores, which indicates, again, progression in subtype 4, and also, I don't show the slide, but we have improved survival outcome for patients in subtype 4, which is a little bit statistically significant, like 0.02 or something. I don't remember the exact subtype. So going back to now that we have these subtypes, which we think sort of capture the diversity, we wanted to use the features of these subtypes to really go back and map that landscape and to really map the patients onto this two-dimensional structure, right? And so for that, we built an elastic net regression prediction model from the proteomic features, the proteins and the phosphocytes, which are represented here by different rows. So the row that's labeled orange is a feature that's used to predict subtype 1. These features predict subtype 2. These features at the bottom are used in multiple models to predict different subtypes. And as you can see, when we plot these features in a heat map across the 210 patients, we see that they primarily cluster according to subtype, with the exception of subtype 3, which you can talk to me about, and I have a reason why that happens that I won't go into. And so that's a 1D representation of the subtypes. Here's a 2D representation on our landscape, again, showing how using these, I think there are 150 some odd, this number, 159 features maybe, map these, we take these patients, we map them to this two-dimensional space using UMAP, and we get this landscape of patient space and relatively separate from each other. These features and define their own biology, so we can do functional enrichment on these features to show. I mean, subtype 2 is pretty distinct with its upregulation of RNA splicing and RNA signaling and downregulation of immune activity, whereas subtypes 1 and 3 have that upregulation of immune activity. Subtype 4 is a little off, and we can also do the same enrichment, look into the biology of the subtypes. At the kinase level, this is using the phosphocytes that are in our elastic net regression model to do and doing enrichment on those phosphocytes. So we're able to look at which kinases are uniquely active, which is indicated in red here, or depleted, which is in blue. And the statistical significance for both figures is indicated by a star. So, now that we have these different subtypes, these proteogenomic subtypes, we wanted to compare them to the mutational subtypes, because as we showed in that bar plot earlier, a lot of AML biology and treatment is really derived on the mutations that are present. So that's almost kind of a different category of subtyping. And if you kind of were paying attention, you notice I didn't include the whole exome sequencing in our proteogenomic subtype definitions for a number of reasons. So this enabled us to kind of have two independent mappings, and we wanted to see how they overlapped. So we looked at the most common mutations in these patients, either individually, such as FLT3 or P53, or in combination, such as IDH1 and 2 and NRAS, KRAS, because we think that these mutations target the same pathway. And you can see, how common these mutations were in the overall cohort, as indicated by the total length of the bar plot. And then you can see how they're distributed across the subtypes by the colors of the individual bars. And so what I thought was interesting was that there was the two subtypes classification systems are kind of complementary, right? So, there's statistical enrichment here and there, but really FLT3 ITD occurs in all the subtypes, right? IDH1 and 2 or DNMT3A occurs in all the subtypes, right? So there's clearly something different going on with the mutations, and the biology is driving these subtypes, which do have biological impact. And so then we wanted to take these and see how they compared with the drug response. So the first thing we can do is, this is how it goes. So we looked at, for example, the FLT3 ITD mutation at NPM1. This is the best annotated mutation that we have because it was measured in the clinic versus in the whole exome sequencing. But we're able to see that many, the presence of a FLT3 ITD variation in the patients is statistically associated with many drug responses, indicated by this plus sign here, whereas NPM1 was not associated. So this is good because this indicates that, okay, if the patient has this mutational subtype, they're preferentially sensitive or resistant to a drug, and we know more information. But there's many drugs out here for which there's no information derived from the mutational subtype. So now we can look conversely at the proteogenomic subtype. So here we have sort of a similar plot, slightly different annotation, I apologize, for the proteogenomic subtypes here, 1, 2, 3, and 4. And what this shows here is if the box is covered, then that subtype is statistically related to the patient's response to, say, venetoclax. So as you can see here, any patient in any subtype has a differential response to venetoclax. If you're in subtype 2 or 4, you'll have a different, you are predicting a different response to NF-kappa B. And if you kind of squint and put these two together, you can see that we're actually adding to the information from the mutation. So we're not replacing it, but we're actually saying, okay, so now mutations give you some set of information, is very valuable. And now the proteogenomic subtypes actually add to that. And then we actually did one more step with our linear models. This is all linear model regression feature selection magic. And that we took the overlap. So we actually built linear models with the combination of subtype and also FLT3 ITD status. So this red bar here means that FLT3D ITD being true alters the state of the predictor in combination with the subtype. So now we can see things like the JAK1 inhibitor shows up as statistically significant. Again, JAK1 not statistically significant until I looked at the slide yesterday. JAK1 inhibitor is not statistically significant based on the presence or absence of FLT3 or ITD1. So we can't distinguish its response. We can't distinguish its response with, where'd it go? Proteogenomic subtype alone, but in combination, we can actually make claims about the behavior of this drug on the patient samples. And I can show what that looks like here, where we can look at subtype 1. For example, again, subtype 1, slightly different response. You have maybe a slightly improved response according to this t-test. This is, I just said this earlier, this is all work by James Pino here. I'll give the acknowledgements later. But James did this very complicated slide, but it does give a lot of information here where we say, look, that if you have a FLT3 ITD mutation and you're given a JAK1 inhibitor, you're actually going to be more resistant to the JAK1 inhibitor if you're subtype 1 than otherwise, but you're going to be more sensitive to the JAK1 inhibitor if you have subtype 2. And again, that change is not statistically significant in the other two subtypes. So it's important, we're kind of showing the importance of the proteogenomic subtype and together with the mutational subtype by the presence or absence of these bars. And the presence of all these blue things mean that combinations are statistically significant, especially like FLT3 ITD and subtype 4 in particular. So given sort of the importance of proteogenomic subtypes and predicting drug response, we started to look at these violin plots where we were able to look at patient drug response. And this, again, is shown by AUC in the ex vivo models, where lower AUC is more sensitive to the drug and higher AUC is more resistant to the drug. And then we can map where the patients fall according to their subtype. And we can, again, James can measure the statistical significance between these different classes. So what we saw with venetoclax was very striking, where subtype 2 patients were much more responsive to venetoclax than the other ones. And this is a drug that Christina called out in the differentiation data as well, the cell state. And then we can compare that to panobinostat. Again, these are things that we pulled out independently without actually reading the Cancer Cell paper because it was still being reviewed and written. But panobinostat patients who belong to subtype 2 actually showed a completely complementary phenotype where they're actually more resistant to the drug compared to subtypes 1, 3, and 4. So this made us think, okay, if we're hitting these patients, we're hitting, if subtype 2 is being hit by venetoclax and all the other subtypes are being hit by panobinostat, maybe we can give them both and they'll hit all the patients. And that's exactly what the data on the plates showed. Because what Christina, when she was describing those plates, they also put common combinations of drugs on there. And this one in particular was rewarding because we're able to look at the distribution of patients and saw that almost all the samples were sensitive to this combination, as would be predicted by this behavior. So we wanted to say, ask if we could use this subtyping, given its power with drug response, to predict drug response in unseen samples. And so for this, we went back to a paper that we also collaborated on, we being the OHSU, PNNL, PTRC, CPTAC folks, and was led by Sunil, where he was able to treat MUL14 cells, which are a FLT3 mutated model of AML, with quizartinib, and model early resistance to the drug as the cells evolved resistance, a late resistance, and then fully resistant. So now we sort of have this gradation. And then again, this stepwise evolution of, well, gilteritinib, we also have quizartinib resistance is in this paper here. So I recommend you read it for all of the amazing data that they collected. I guess. But this allows us to really have these cell lines that are distinct based on drug treatment, that we can then use to, and we also have proteomic and phosphoproteomic data, so we can actually easily apply our subtyping model to these cells. So when we first did this, it was a little underwhelming because the cell lines are all clustered sort of in the middle here of this landscape. And this is not incredibly surprising because cell lines are all genetically identical to begin with. And so you can tell that the drug treatment didn't move them across this landscape in a large way compared to the original patient diversity. However, we were able to sort of predict the subtypes, and we did find different subtypes. So we found that, the drug naive samples were primarily subtype 2, all subtype 2 to begin with. And as they gained resistance to quizartinib, they migrated from subtype 2 to other subtypes. However, despite this sort of shift in this direction, we weren't able to really predict drug response as strongly as we would have liked. So for that, we went back to another project that we did together. This is, again, the OHSU PNL folks, where we did explicit predictive modeling, proteogenomic modeling of drug response using linear regression models. And this paper here that came out, God, was it this year? Earlier this year. It's been a long year. Which combined proteogenomic data and drug response and really experimented about the power of drug response prediction using, this is just a 38 patient cohort, not even the 210, but we looked at genetic mutations, transcripts, the combination of transcripts and proteins, the combination of proteins and phosphatides, sort of every combination of data to see how well, using sort of held out data, the predictions were able to correlate with the original drug response across various drugs. I think 24 or 26 drugs. And what we showed here is with elastic net, lasso regression, logistic regression, at least with the two continuous regression models, proteins were very successfully able to predict drug response. And so we were confident that we're able to use linear modeling in this larger cohort to predict the drug response. Oh yeah, I forgot about this one. So this one shows that not all models are predicted equally. This is actually in our larger cohort, but venetoclax, which is one of the drugs we're interested in, was a very strong predictor. Panabinostat is also pretty high performing, but before you go off and try to predict proteogenomic, drug response from your proteogenomic data, I like to give this slide as a caveat, say it might not work, but a lot of drugs do work, and I like to be upfront about that because some models perform better than others. The other thing that we're able to take out of this is the beauty of linear regression is that the features are fully extractable, so we can look at the features identified in each model. And this paper showed that they are dysregulated in resistance, drug resistance. So there's this, we know that the features that are selected are also biologically important. So what we did is we built predictors from the larger data set now, predictors of venetoclax and panabinostat, took the features and then used them to map a new landscape of drug response. And so the features here now are less likely to cluster by subtype because they're not selected for subtype classification. They're selected for panabinostat and venetoclax drug response. So you can see that now we have less distinction than we had earlier. And you can see that again in 2 dimensions here, but they do tend to segregate by drug response, right? So now we have panabinostat responding patients on the left, venetoclax responding patients on the right, and those that are sensitive to both are in green. These are using kind of arbitrary cutoffs for sensitivity, but give you a general sense of how the features that you use to map the landscape shift the landscape, right? So here we're now specifically targeting drug response rather than overall tumor biology. And so we're able to now use this model to predict drug response in these cell lines. And what we showed here is that the area under the curve, the predicted area under the curve by the model tended to decrease as the cells became more resistant to quizartinib for panabinostat. So panabinostat becomes more able to, the cells die more with panabinostat as they become more resistant. So they're resistant to quizartinib, but more sensitive to panabinostat. but actually go in the opposite direction for venetoclax. So as the cells become resistant to quizartinib, they're also becoming resistant to venetoclax. And then Sunil went back to these same cell lines that he painfully grew while he was doing his PhD. He's now a postdoc, and he was able to validate these areas under the curve, at least the shifts. Actually, the results were much more striking in person than in our model, which again was really rewarding because now we have essentially, you know, for quizartinib resistant cells, we have another drug that seems to be which the cells are quite sensitive to. So that was pretty exciting from the clinical standpoint and from the modeling standpoint, where we actually do want to build biologically and clinically relevant models. So that's where the paper stands right now. We're wrapping everything up for submission, but I wanted to sort of drill home this idea a little bit further, that with proper model systems, and this is really the drug sensitivity assay, the ex vivo sampling, which allows us to measure the patient tumor using all the omics measurements to provide a mapping of the different patient samples to this landscape. And then measuring the drug response allows us to better understand and identify these potential relationships between proteogenomics and drug response. So here's our acknowledgement slide. Again, Sunil Joshi was a student of Ailey Trayer's. So Ailey they did a lot of the early resistance work. I didn't actually include everyone that was in Christina's paper. I apologize for that. And then the work on the proteogenomic stuff is led by James Pino and Camilo Pozo, who have done, Camilo did all of the NMF clustering and a lot of the elastic net regression modeling for subtype prediction and classification. James Pino has led the paper effort as well as the prediction of the proteogenomic prediction models. And then, you know, everyone else included, Karen is the PI, Brian and Jeff are the PIs of the OHSU work. It's been a truly collaborative effort. Is there anyone else you want to call out, Christina?
No, I mean, I obviously kind of acknowledge the fact that for the Beat AML work, it was a massive village that really tried to that helped with that effort, and it's ongoing. So, you know, I want to also thank a shout out to the patients who donated all the samples. We tend to like to do that because it is really important for our research. And then another shout out to all the patient sample processors in our team that actually process all the samples for us to do this work. It's a monumental effort. So, yeah, just happy to take any questions.
Awesome.
And I guess maybe one more thing I want to say is, you know, the collaboration between PNNL and OHSU has been super fruitful. And I know that there's, you know, you know, we are across the university are trying to develop this tight collaboration between our two groups, and want to definitely express how wonderful a collaboration we've actually had with this UO1 grant and the opportunities that we've been afforded across the group. I know there's a number of other additional pilot project awards and other awards that are available. Maybe, you know, I don't know if Liz and Laura are going to talk about that later, but it has been a very fruitful collaboration. I would encourage people to seek out funding opportunities and projects. So.
Thanks for that, Christina. And I see a hand up raised. Let's see, Indra Mealy, if you want to just go ahead and come off mute and ask your question.
Yes, thank you. You know, first, kudos as a fellow ex-Canadian, kudos to the team and Christina and Sarah for a real tour de force. Very impressive. I guess what struck me is in the first half you mentioned the 6 cell states. And then we have these 4 proteogenomic subtypes. So how do those relate to each other? And I'm assuming the proteomics was all done in bulk measurements. So how can you deconvolute that?
So that's a good question. So I have another preprint which tries to answer this question of proteomic deconvolution using single cell RNA markers. And I don't think that problem has been effectively solved, right? Taking gene markers from transcripts and mapping them to proteins. I can send you, the preprint's really just a framework for evaluating that question because we didn't have data. But we did do this analysis, and we do have that preliminary analysis for these data sets. So now we can map the 150 subtype, the 159 patients with that have RNA-seq into subtypes. And we do see enrichment in that, what's the word, premature, differentiated, what's the word I'm looking for, state in subtype 2. So the patients that are sensitive to venetoclax, the subtype 2 proteogenomic subtypes are certainly enriched in that, I don't know, the more stem-like patient samples, but they don't have the 6, right? We don't have that level of granularity at the proteome, and I don't know if that's a data thing or a deconvolution thing. And it's something that we're actually looking at in the next PTRC, is actually trying to build better protein markers of cell type so that we can better deconvolve proteomic data in the environment. Christina, do you want anything to add?
No, I think you did a good job of
Okay.
explaining that. Yeah, yeah, definitely.
Forget words.
And what was also striking, if I could ask another question, is the pair 1, that marker. That sounds some interesting biology. How does that relate to the proteogenomic subtypes?
We haven't really looked at pair 1 at the protein level. Like it didn't, we have it as sort of an action item, but we haven't really looked as far as a marker goes. I don't know if it's a marker at the protein level. I know it's in the proteins. We've measured it, but I haven't looked at how
it's associating with the subtypes. Yeah.
Yeah.
No, I think that's a big piece of the work that we want to do.
Yeah.
We're doing some validation work right now in AML cell lines. We know it's expressed at the via Western blot across all kinds of AML cell lines. And then obviously we're going to dig into the phosphosites and the total peptides that are identified in the cohort here. I don't think it's expressed on all of them, because otherwise it would have shown up in our basic spreadsheet. So I think we need to do some digging into that data a little bit more, but we're definitely interested in looking into that. We think it's going to be some type of biomarker of response. I mean, it is potentially targetable. It is a possibility. It's got downstream activation of things like SYK and different pathways downstream that could be targeted. We've looked into the thought of it being a potential ADC target, but it might be expressed on other tissues, which might make it less viable for that. So yeah, definitely a lot of interest right now in the lab in validating and understanding the biology of pair 1.
Great, thank you for that.
Great. I see next a hand up by Brian Carberg. If you want to go ahead and unmute.
Oh, hi. Yeah, I'm Brian. I'm a PhD student with Kyle Elrott at OHSU. My question is about the choice of the non-negative matrix factorization as the unsupervised clustering method. Like, how was that chosen as the method, and are there other options to benchmark that against? It sounds like that method kind of had an inbuilt iterative process. And then relatedly, in terms of those labels that are assigned to each sample, is it possible that some of these samples are expressing or exhibiting multiple characteristics of multiple subtypes? Could the problem be structured as a multi-label prediction problem?
A lot of questions. Okay, so I'm going to start with the choice of NMF, and I'm going to be honest with you, it's been pretty widely used in the proteogenomic field as a way to integrate these data, and I've been pretty pleased with the results, both because, again, it's biologically interpretable. We can benchmark it. There's a co-phonetic correlation that determines how well our clusters are working, so that was really kind of nice. It also behaved pretty well with the different scales of data. There's some normalization and obviously non-negativization that had to go into this, but it was, and Camilo did a lot of that work to really just dive into the paper and move our stuff over. It didn't behave super well for the genetic data. So we pulled it, and that was the big downfall. So I didn't really look at other unsupervised approaches because we were pretty happy with these. And a lot of things I do worry about how much we're going to put, sort of double dip for proteins and phosphosites and transcripts from the same gene, right? So it was really helpful, and it's also very extensible for metabolomics. People incorporate that, and they put all of the omics into one non-negative matrix and they cluster. So I've been pretty happy with that. And as far as the multi-label prediction problem, I mean, ostensibly we did do that. So we what I kind of hid is that we did the clustering on 159 patients because that's the ones for which we had all the data. And then we did a regression to basically from just the proteomics data, predict back the patients. And we're able to do that with the proteomics and peptide phosphoproteomics completely accurately for the 159. And then we projected them to the 70, the rest. Can't do math anymore. And so that was pretty successful, but we actually, in our supplemental data, we actually experiment with lots of different data modalities. Subtype 3 was very reliant on the RNA-seq. And so that's why it was kind of scattered throughout when we did purely protein mapping, because it wasn't as captured. So I think I like that it was able to capture different data modalities and different subtypes. Yeah, again, I'm happy to send you the details about the methods. I think it's really helpful, but it's not super, it's not plug and play.
Thank you.
Is there any other questions from the field? All right. Well, thank you so much to Sarah and Christina. This was a fantastic seminar, and thanks for the claps. Yes, and we will be posting this on our PMedic website, so anybody can access this again and click those QR codes for all these great citations in the YouTube video that we'll have posted. Thank you all so much.
Yeah, thank you.
Thank you so much. Have a great night.
Yeah, bye-bye. Yeah.